expressions you need to know. First, there are 3 planes of section which are used when cutting through the brain. Dividing the brain in two along the midline, the natural division between the two sides, produces the sagittal plane. Cuts made parallel to this are called parasagittal sections. At right angles to this, running from top to bottom, is the coronal plane. The third axis, which passes through the brain from side to side, is the horizontal plane. To describe the relative positions of different structures, a superior structure is closer to the top of the brain than an inferior one, like the brainstem. Anterior structures are closer to the back of the head than posterior ones, like the cerebellum. For the remaining terms, you need to be facing the front of the brain. Medial describes a region close to the midline, and lateral refers to one further away from it. On the outside, you're looking down on the front of the human brain. From here, the most striking features are the 2 cerebral hemispheres. However, a number of important structures are found below the cerebral hemispheres, at the back of the brain. The largest of these is the cerebellum. This literally means little brain. It's not a bad name for this complex structure, which is involved in a large number of functions, mostly to do with motor coordination, balance, and the maintenance of posture. Just in front of the cerebellum is the pons, named from the Latin, as it appears to bridge the 2 halves of the cerebellum. Passing through the pons are bundles of nerve fibers ascending to and descending from the more superior parts of the brain. Anterior to the pons is the pituitary gland, important in controlling the activities of the endocrine system. The most inferior part of the brainstem is the medulla. It contains large numbers of axons. These relay information between the brain and the spinal cord. It also contains nuclei, that is, collections of nerve cells, which are responsible for the regulation of heart and breathing rates. This is the region which has been damaged when a person is certified as brain dead. Above the brainstem, and hidden by the cerebral hemispheres, is the diencephalon. It has extremely important and widespread connections with other parts of the central nervous system. Part of it is formed by the thalamus, which has a key role as a relay for inputs to the cerebral hemispheres. The most characteristic feature of the cerebral hemispheres is the way their surface or cortex is organized into numerous folds. The cortex consists of layers of nerve cells. The folds increase surface area, which means that more nerve cells can be accommodated. Each ridge is known as a gyrus, plural gyri, and each depression as a sulcus, plural sulci. The precise pattern of these folds varies considerably between individuals and even between the 2 halves of the same brain. However, some principal gyri and sulci can always be identified, and specific behavioral functions have been related to them. One fold in the cortex is very obvious. It is known as the lateral fissure. Below it is the temporal lobe. Here, along the line of the lateral fissure, the primary auditory cortex is located. Another feature which can always be identified is located halfway along the lateral fissure. This is the central sulcus. The gyrus in front of the sulcus is the precentral gyrus, the location of the primary motor cortex. Behind it is the postcentral gyrus, the site of the primary somatosensory cortex. Fibers carrying information from the body about sensations such as pain, temperature, and touch terminate here. This organization is the same on both sides of the brain. In front of the central sulcus and above the lateral fissure is the frontal lobe. It's here that the motor cortex is located. But it also contains a large area of association cortex. It receives inputs from and sends outputs back to the other parts of the cortex. There are clearly defined areas implicated in receiving and processing information. Take visual information. This is processed at the posterior part of the cortex, the occipital lobe. The cerebral cortex is the destination for incoming sensory information. Here the information is analyzed, and if necessary, a response is initiated. The brain is metabolically very active, and it needs a plentiful supply of oxygen and glucose for energy. These are carried in the bloodstream. The brain is supplied by an intricate network of arteries. Many are visible on the surface, but branches ramify deep into the cerebral hemispheres and cerebellum. Going in. Successive slices through the superior part of the cerebral hemispheres show their internal structure. These are horizontal sections photographed one after another. The narrow gray margin around the edge is the cerebral cortex, the gray matter. It is composed of millions of nerve cell bodies. This is a light microscope section through the cortex, prepared using the Golgi method. There are several different cell types, which are identified by the differences in cell shape and size. The cerebral cortex can be subdivided into 6 layers based on the distribution of the different cell types. The outer layer, layer 1, has very few neuronal cell bodies. Instead, it consists of a vast network of axons and dendrites, forming synaptic connections with each other. Layers 2 and 3 contain a variety of cells whose axons project to other areas of the cerebral cortex. Layer 4 mainly contains stellate or star-like cells. This is the layer where all inputs to the cortex terminate. Layer 5 comprises large pyramidal cells. These are the principal projection cells of the cortex, sending their axons either to other areas of the cortex or to other parts of the central nervous system. Layer 6 contains a variety of cells. Axons from these cells may project either to other areas of the cortex or to other parts of the brain. Beneath the cortex is the white matter. This is composed of millions of axons. Most of these are surrounded and insulated by a fatty sheath of myelin, here stained black. In life, this sheath is white, which gives the white matter its characteristic appearance. The sheaths are not continuous. There are gaps where there is no myelin. These are the nodes of Ranvier, where the action potential is regenerated. The axons include projection fibers traveling to and from the cortex, and association fibers connecting different regions of the same hemisphere. This photographic journey through the brain was achieved by cutting off successive slices of 40 microns.
You know so much white
It gives the
matter.
impression of traveling deeper into the brain. You get a very strong idea of how bilaterally symmetrical the overall structure is. The two hemispheres are linked by a bridge of fibers, which appears about 2 centimeters down from the top of the brain. This bridge of white matter is called the corpus callosum. It is formed by axons which link corresponding areas of the two hemispheres. On each side of the corpus callosum, dark, almost black areas appear in the hemispheres. They are fluid-filled cavities called the lateral ventricles. They are the remnants of a hollow tube from which the brain develops. The whole ventricular system has a complex shape. The two lateral ventricles constitute the greatest part of the system. Typically, they extend a long way forward within the cerebral hemispheres. The lateral ventricles are filled with a cerebrospinal fluid, which acts as a physical support for the brain and the skull. It also carries nutrients and waste products of neural metabolism. It is continually produced in the lateral ventricles, and from there is forced into the narrow midline cavity, the third ventricle. From here, it flows down the tube-like cerebral aqueduct in the midbrain to the fourth ventricle. The fourth ventricle is located between the cerebellum and the medulla, which narrows to become continuous with the central canal of the spinal cord. From here, some of the cerebrospinal fluid goes down the spinal cord. The rest spreads over the surface of the brain to bathe and protect it.
Now that was the anatomy.
Systems of the brain.
This is the physiology now, what these parts of the brain actually do. These are the systems. And those are starting at number 21.
It's perfectly possible to see the brain simply as collections of individual cell groups and fiber bundles. But a better, more informative approach is to look at it in a more functional way, to see how the different components work together in the systems of the brain. From now on, we're going to look at some of these systems and try to make sense of structure in terms of function. Thalamus. The cerebral cortex processes and analyzes sensory inputs and initiates motor activities in response to those inputs. How does the information arrive here? Deep within the brain, next to the midline, is the thalamus. It's described as a single center, but in fact it consists of two structures, which are actually collections of about 30 different nuclei, each of which has its own specific set of connections with other parts of the brain. A major function is to relay sensory information to the cerebral cortex. All the primary sensory inputs, except smell, are routed through a specific nucleus. Each lateral geniculate nucleus receives information about sight, which is then relayed to the primary visual cortex. Each ventrobasal nucleus receives somatosensory information and relays it to the primary somatosensory area. Others, the ventral nuclei, relay inputs to the motor areas of the cortex. How does this information get from the thalamus to the cortex? Neurons within each specific nucleus receive incoming sensory information which arrives on their dendrites. If sufficient excitatory impulses arrive at the same time, the thalamic neuron generates an action potential. This is then transmitted along the axon, which terminates on neurons in the cerebral cortex. The thalamus also contains a group of non-specific nuclei. These modulate the activities of the specific nuclei, such as the lateral geniculate, the ventral basal, and the ventral nuclei. The nonspecific nuclei serve to raise or lower the threshold level at which information is sent to the cortex. Many nuclei appear to be interconnected, and most receive inputs from the region of cortex they send axons to. In fact, the thalamus can be considered as the gateway to the cortex. Visual system. The Clothed Maha by Goya. But what is happening when we look at a painting like this? How does the information about a pattern of colors on a piece of canvas get to the brain? The painting reflects light waves, some of which enter the eye. They are focused by the lens and arrive at the retina, where they form an image. Because of the way light travels through the lens, this image is back to front and upside down. The retina is an extension of the central nervous system and consists of layers of nerve cells, behind which are photoreceptors. They convert light energy into electrical potentials. There are two types of receptors: the larger rods, sensitive to low light levels, and the cones, which require higher light intensities and pick up color. The electrical potentials generated by the rods and cones are relayed via a layer of bipolar cells to the retinal ganglion cells in the outer part of the retina. These are the only cells in the retina with axons, which transmit action potentials. The retinal ganglion cell axons come together at the center of the retina to form the optic nerve, which carries the visual information to the brain. The actual information which will be received by the brain is the result of complex interactions between the inputs from the two eyes. From below, we can see how this works. Information arriving in the half of the retina closest to the nose, the nasal retina, is segregated from the information arriving in the half of the retina closest to the temples, the temporal retina. At the back of each eye, the fibers from the nasal retina are separated from the fibers from the temporal retina within each optic nerve. The optic nerves enter the skull and pass back beneath the brain to come together at the midline to form the optic chiasm. It's a crossing point. The axons from the retinal ganglion cells in the nasal half of each eye cross the midline, while those from the temporal half do not. Approximately half of the retinal ganglion cell axons, those from the nasal half of the retina, cross over, so that all the fibers from the right half of each retina go to the right side of the brain, and vice versa. The bundles of fibers going to each side of the brain, the optic tracts, travel backwards under the cerebral hemispheres to terminate in the two lateral geniculate nuclei of the thalamus. A coronal section of the lateral geniculate nucleus shows on close examination that it is a six-layered structure in which the axons of the retinal ganglion cells from each eye terminate in alternate layers. So the layers colored black receive inputs from one eye, while the white layers receive from the other. The cells of the lateral geniculate nucleus send their axons via the optic radiation to terminate in layer 4 of the primary visual cortex in the occipital lobe. The cerebellum has been removed to show the path more clearly. The inputs from the two eyes terminate as alternating bands about 0.5 millimeter wide, and the cells within each band respond specifically to inputs from one eye. These bands form the basis of the ocular dominance columns. The nerve fibers terminating in the primary visual cortex are not distributed in a random order, but follow a precise point-to-point mapping. The inversion of the image on the retina, together with the crossover of nerve fibers at the optic chiasm, produces a representation which is inverted, with the right half of the painting represented in the left hemisphere and the left half in the right hemisphere. Information from adjacent areas of the retina is represented over adjacent areas of the cortex. In other words, the axons of the visual pathway are topographically organized. The actual representation of the image on the cortex is distorted because some parts of the retina have a larger area of representation than others, and the cortex is a highly folded structure. Somatosensory system.
Now this is called a homunculus, this model here. Kind of a strange looking.
This is a representation of the male human body, but it is not lifelike.
Thank God.
The model is based on the way the body's surface is represented on the primary somatosensory cortex. This is the area of the cortex which receives information about touch, temperature, and tissue damage, which is perceived as pain. The odd proportions illustrate the way that regions which have a dense sensory innervation, such as the lips and fingers, are represented over a greater area of cortex than poorly innervated regions, such as the shoulder and upper arms.
So your lips have as much innervation as your arm does.
How does the sensory information get from, say, the thumb to the cerebral cortex? Information about touch, temperature, and tissue damage all travel together from the sensory organs via the peripheral nerves to the spinal cord, which they enter via its dorsal roots. In the spinal cord, the nerves divide into two groups, those carrying information about tissue damage and temperature, and those carrying information about touch. A transverse section. Fibers carrying information about tissue damage and temperature, here in purple, terminate on cells located in the outer part of the dorsal horn of the gray matter. The axons of these cells cross the midline and ascend in the spinal cord within the white matter. This is the lateral spinothalamic tract. There are thousands of axons in a tract. The fibers which convey touch information do not terminate in the dorsal horn. They stay on the same side of the midline and ascend in the white matter straight up the spinal cord in two bundles known as the dorsal columns. This whole arrangement is mirrored by nerves originating in the other side of the body. At each level of the spinal cord, more incoming sensory axons join the fiber tracts. They ascend to reach the brain within the skull. Hidden beneath the cerebellum is the medulla. Here, the axons of the dorsal columns terminate on two groups of cells called the dorsal column nuclei. The axons of the cells in these nuclei cross the midline and ascend through the brainstem to terminate in the ventrobasal nucleus of the thalamus in the diencephalon. The axons of the spinothalamic tract, which crossed over in the spinal cord, travel straight through the brainstem and also terminate in the ventrobasal nucleus. All the somatosensory information from one side of the body projects to the other side of the brain. As we've already seen, the thalamus can be described as the gateway to the cortex. The axons arising from the cells in the ventrobasal nucleus transmit all these different types of information to the primary somatosensory cortex. The primary somatosensory cortex can be readily located because it forms the postcentral gyrus. All the inputs terminate in layer 4 of the gray matter. The organization of these inputs within the primary somatosensory cortex is highly specific. Each part of the body is mapped to a particular region. Information from the foot arrives near the midline, and information from the face arrives on the lateral part of the hemisphere. The oddly shaped sensory homunculus is based on this map.
There's also a homunculus for the motor strip as well. This is the somatosensor.
Pyramidal tract.
You know, these are the motor tracts.
Layer 5 of the cortex, the principal projection layer. Motor outputs originate from this layer of the cortex of the precentral gyrus, the primary motor cortex. Like the primary somatosensory cortex, it too has a topographic representation of the body. But those of the sensory system are different to the proportions of the motor system. This reflects the range and precision of movements performed by those parts. How does the information from the primary motor cortex get to the muscles which it controls? The brainstem, midbrain, pons, and medulla serve an important function in the routing of information from the cortex to the rest of the body. From layer 5 in the primary motor cortex, the axons of the pyramidal cells descend past the thalamus and through the midbrain. Below that point, they are located beneath the surface of the ventral part of the pons. Below that, they run down the ventral surface of the medulla in 2 discrete bundles, one on each side of the midline. A transverse section through the medulla shows these fiber bundles as very prominent triangular shapes. These are known as the pyramidal tracts. At the end of the medulla, the fibers of the 2 pyramids cross the midline and descend on the opposite sides of the spinal cord. When the appropriate level of the spinal cord is reached, the axons terminate in the ventral horn. Some terminate on interneurons, which in turn project onto motor neurons. Others terminate directly onto motor neurons. This is obviously a model, but there is good correspondence in overall shape with a transverse section. The ventral horns stand out from the more grainy-looking white matter. They are densely populated by the dark cell bodies of the motor neurons. The axons of these motor neurons emerge from the spinal cord via the ventral roots to project onto the limb muscles. Outside the spinal cord, they join with the sensory axons to form the peripheral nerves. Basal ganglia. The output from the primary motor cortex is under considerable regulation from other centers within the brain. An important source of influence is a group of nuclei known as the basal ganglia. They comprise a number of discrete nuclei which have key roles in the initiation and coordination of motor activities. There are 4 components: the substantia nigra, the globus pallidus, the pitamen, and the caudate nucleus. They are shown as 4 separate nuclei and were originally described that way. But in fact, they're intimately related in functional terms. Although they're implicated in motor control, they don't have a direct output to the neurons in the spinal cord. Instead, they influence the output via the ventral thalamus. A coronal section. This shows some of the pathways between the centers, the thalamus, and the cerebral cortex. It's complicated. Here's a simplified model. Inputs from sensory and motor areas of the cerebral cortex are received in the caudate and putamen. The principal output is via the globus pallidus to the ventral nuclei of the thalamus, which in turn project back to the motor and premotor areas of the cerebral cortex. An additional output from the nuclei terminates within the substantia nigra. The main characteristic of control systems, this one included, is the connection of a series of centers in a loop, allowing for feedback control. A good illustration of the operation of such feedback loops is the failure of the substantia nigra. Photographs of successive coronal sections of the brain. The substantia nigra is located in the inferior part of the central mass of the brain. Substantia nigra literally means black substance, and its telltale color can be picked out. The dark color is because it contains a large number of neurons that utilize dopamine as their transmitter. Here, the section has been stained for the dopamine precursor enzyme. It shows the high proportion of cells that contain it. These dopamine-containing cells can die, so the vital feedback pathways to the other nuclei are lost. The result is a loss of motor control, muscle tremors, and difficulty in initiating movement. This is Parkinson's disease. Cerebellum. Important regulation of motor activities comes from the cerebellum, the highly folded structure at the back of the brain. In a parasagittal section, the extent and intricacy of these folds is clearly visible. In some ways, the cerebellum resembles the cerebral hemispheres. Like them, it has an outer cortex of nerve cell bodies, under which are myelinated fibers. The cerebellar cortex also has a laminar structure, but of 3 layers instead of the 6 of the cerebral cortex. The 3 layers are an outer layer of cellular processes, a middle layer of Purkinje cells, named after the neuroanatomist who first described them, and an inner layer of small interneurons. The most important is the Purkinje cell layer. It is 1 cell thick and is composed exclusively of Purkinje cells. These are the most prominent cells of the cerebellar cortex, and they're the only cells whose axons project out of the cerebellar cortex. This is a drawing of a typical Golgi-stained Purkinje cell. It has a large oval-shaped cell body, and an extensively branched dendritic tree emerges from one end. If viewed from the side, Purkinje cells look very thin because the dendrites only branch in 2 dimensions.
So those are fan-shaped. They branch.
The axon, labeled A, emerges from the cell body. All Purkinje cell axons terminate in nuclei deep within the cerebellum. Axons from these nuclei project to other parts of the brain to influence motor activity in the body. The cerebellum is involved in acquiring skilled movements which may become almost automatic with practice, such as the maintenance of posture, walking, or playing a sport. To provide this control, the cerebellum receives sensory inputs directly from the muscles and joints and balance organs, but also via the motor and sensory areas of the cerebral cortex. Some of its output is directed back to the cerebral cortex via the ventral thalamus, while the remainder is routed to the motor neurons in the spinal cord. Superior colliculus. With new stimuli at the edge of the visual field, we turn our heads. This is due to the superior colliculus, These two small bumps are located on the dorsal part of the midbrain, one on each side of the midline. They're completely hidden by the cerebral hemispheres. The main input to the superior colliculi comes from the retinal ganglion cells, particularly those at the edges of the retina. The axons of these cells divide, and one branch goes to the lateral geniculate nucleus of the thalamus. The other continues over the surface of the thalamus to terminate in the superior colliculus. Inputs are also received from the auditory and somatosensory systems. We've already seen how information about touch arrives at the thalamus. It comes from fibers ascending via the dorsal columns from spinal cord to thalamus. These fibers give off branches which terminate at the superior colliculi. The principal output from the superior colliculi is to motor neurons in the upper levels of the spinal cord, which project to the neck muscles to turn the head. Control of these movements does not originate from the motor cortex. Patients who are blind as the result of damage to the visual cortex may still turn their heads towards flashes of light, although they are not actually conscious of having detected the flashes. So their superior colliculi are still functioning.
It's something, even though they don't see the light, they'll turn toward it. Something, isn't it?
Hippocampus.
If their optic nerve is intact.
Deep within the brain are the lateral ventricles. Lying alongside them is the hippocampus. In evolutionary terms, it is the oldest part of the cerebral cortex. As the newer part expanded, the hippocampus became folded under the temporal lobe. Here it is associated with the gray-colored fornix. The folding has resulted in a curious Swiss roll-like appearance. The structure has two parts, the hippocampus and the inner dentate gyrus. These are differentiated mainly by cell type. The hippocampus consists of only 3 layers. The principal cell layer is sandwiched between 2 layers of axonal and dendritic processes. In the hippocampus, the central cell layer contains only pyramidal cells. These are the projection cells, and this layer equates to layer 5 in the rest of the cortex. The dendritic processes of the pyramidal cells extend into layers 1 and 3. The central cell layer of the dentate gyrus consists exclusively of granule cells, which are typified by having dendrites, which emerge only from one end of the cell body.
They're very similar to Purkinje cells, granular cells, very similar.
The majority of axons from these pyramidal and granule cells terminate locally within the hippocampus, although some project to the nearby cerebral cortex. In functional terms, the adjacent cerebral cortex is usually considered along with the hippocampus. It's known as the entorhinal cortex and lies along the inferior surface of the temporal lobe. Together, they are known as the hippocampal region. The principal inputs to the hippocampal region come from the sensory and association areas of the cerebral cortex, and it sends some fibers back to these regions. Other outputs from the hippocampal region emerge from the superior aspect of the hippocampus and pass upwards and backwards to form a bundle of fibers called the fornix. This passes forwards over the top of the third ventricle, seen more clearly if one of the lateral ventricles is removed. The fornix isn't a nucleus. The fibers terminate in the anterior part of the thalamus and the mammillary bodies, which are part of the hypothalamus. The hippocampal region appears to be important for learning and memory. Patients with damage to the hippocampal region on both sides of the brain are unable to learn new facts or remember recent events, although their recall of more distant events is usually unaffected. Hypothalamus. The thalamus. Below it is located the hypothalamus. Like the thalamus, it consists of a collection of nuclei which are mirrored across the midline. In addition to projections from the hippocampus, the hypothalamus receives a large number of inputs from various parts of the nervous system. These include inputs from the viscera, projections from neurons in the spinal cord and brainstem, fibers from the cerebral cortex, some fibers from the thalamus, and also a small but significant input from the retina. With the exception of the retina, all these regions receive outputs from the hypothalamus. One of the most important outputs from the hypothalamus goes to the pituitary gland, which hangs down below the hypothalamus. The so-called hypothalamo-pituitary projection consists of two different pathways. In the first, some hypothalamic neurons send their axons directly to the posterior part of the pituitary, where they terminate in the vicinity of fine capillaries and release hormones, which are important in the regulation of fluid balance and blood pressure. In the other pathway, the axons of hypothalamic neurons terminate within a special capillary network at the base of the hypothalamus and release hormones, which are then carried in the bloodstream by vessels in the pituitary stalk, which link the hypothalamus with the anterior pituitary. The hormones transported by the so-called pituitary portal system regulate the synthesis and release of hormones by the cells of the anterior pituitary. These hormones are important in reproduction and in the regulation of general metabolic activity. Reticular system. The brainstem. Its core is formed by a number of nuclei, which are known collectively as the reticular system. These nuclei contain neurons with extensive dendritic branches. This is a drawing of a transverse section through a brainstem. It shows several typical reticular neurons with horizontally radiating dendrites. Most ascending and descending axons give off side branches to the reticular system. They terminate as synapses on these dendrites. It's not a well understood system, but what we do know is that the inputs are extremely diverse, and the cells of the reticular system project to virtually every area of the brain. The activities of the reticular system are as varied as its connections. By relaying information to the Purkinje cells in the cerebellar cortex, and by sending outputs to the motor neurons in the spinal cord, it is involved in the coordination of motor activities. Other neurons have axons which form part of the autonomic nervous system. They help to regulate heart and breathing rates. An important output of the reticular system forms part of the pathway known as the ascending reticular activating system. Neurons in the brainstem reticular formation project to the nonspecific nuclei of the thalamus, which in turn project to all areas of the cerebral cortex. Modulation of activity within the reticular activating system alters the state of alertness of the individual. It helps to entrain the sleep-waking pattern, to concentrate the mind on a particular activity, like reading a book, or heighten awareness of new stimuli, like pain. Damage to the neurons, which give rise to the ascending reticular activating system, results in coma. Brain journey. To conclude, successive photographic sections down through the brain.
Now the way they make this is they take a brain and they freeze it in liquid nitrogen, and then they cut it in 40 micron slices. 40 microns is 41 thousandths of a millimeter. They photograph each one separately, and then going at about 16 frames a second, or 16 slices a second, you get the appearance you're just going deeper within the brain. Notice how much white matter there is. Your brain is mostly white matter. You hear about the brain being gray matter, but it's mostly white matter. And there you can see the ventricles of the brain. Those are the lateral ventricles. Now the gray matter that will appear down in here, that's the basal ganglia. These are basal ganglia. That's the putamen, caudate nucleus, and right up there is globus pallidus. Substantia nigra is further back here. Back here. And the connection between the hemispheres is the corpus callosum, which is right there. You can see the cerebellum starting to appear back there.
How do they do this?
They freeze the brain in liquid nitrogen and then slice them off in slices and photograph them. That's what they do, one by one. You're looking at about 16 of them every second, 16 frames a second. That's a lot of slices. Very thin slices.
What do they use for the precision slices like that?
What's that?
What do they use for that precision slices like that?
They have a device, a microtome, has a little, it's kind of like shaving off, you know, if you ever been to the old meat store where they shave off like some bologna real thin, that's what it does. It's kind of like real thin slices of like a ham or something. It's similar to that, that's what it is. Put the brain up there and start slicing it off. A little bit more precise than the butcher shop. But, so.
A final word. Much of the way the brain is described, the use of unusual expressions and terms, and the apparently arbitrary subdivisions that have been made, are relics of the past, derived from the days of the early neuroanatomists who were describing what they could see. Despite the arcane nature of these terms, they remain the starting point for fruitful study, the understanding of how the components of the brain are related in functional terms.
So these terms that are used here, a lot of them have been around a long time, like globus pallidus and the putamen, and those have been around since really the 1400s. Those are very, very old terms. Most of them are Latin terms, some are Greek, most are Latin. And so that's why they're kind of strange sounding names. They're just really, really old names. All right, I tell you what, we're going to take a break here in a minute. Then when we come back, we're going to take a look at the spinal nerve and the spinal cord model and go through that. And then we're also going to look at the eye model and talk about the visual pathway. And then I think after that, I'm going to set up kind of like a practice quiz for you on some of this stuff, and you can start taking a look at that. All right, so have any questions on anything before we take a break? Anything about the video? It's a really good video. The narrator's a little monotone at times, but it is a very good video. It has lots and lots of good general information. And as I said, on pages 17 and 18 is the stuff I want you to know about. So, anyway, all right, we'll go ahead and take a break. We'll come back and look at the spinal cord and the eye model.
What was the name of the video?
What's that?
What was the name of the video?
Brain